Hello again, everyone. This is again Pawa, and we are going to now go through biology for the first part. So I'm just going to cycle through biology and then chemistry and then physics, and we'll keep on going like that. So, like I said last time, the for these sessions, it's just going to be on middle school level biology. I'll try to make them as engaging as possible. If you ask me, high school is where it gets super interesting, but we still need the fundamentals required in middle school. So, for this session, we're going to look at biology. So just to start, hmm, okay. So we're going to look at what are the fundamentals of biology, right? Now remember from what we discussed last time, biology is essentially the science of life. It's the study of life, right? So questions like what constitutes something that is alive? What are the characteristics required for life? That's what we're going to cover in biology, right? So if you've ever been curious about stuff like when is something considered alive, or what does it mean to actually be alive, but in a physical sense, not necessarily philosophical, that's what biology looks at. Um, Now, that is not to say that there could be some alien out there in another galaxy or like some kind of planet that's really far away that has different features of life. But the biology that we are studying is to do with what we know so far of the life on our planet, assuming there even is aliens out there. Okay, now the best place to start in biology is to do with cells. Okay, now the word cells itself is just taken from normal English just to mean a single block of something, right? And this was a term that was popularized by Leeuwenhoek. I think I'm butchering his name, but I think his name was Robert van Leeuwenhoek, something along those lines. But he was a scientist who invented the microscope, and he used the microscope to be able to zoom in on these very tiny animals, upon which he could notice that those animals were made up of these very small components, which he called cells, right? But he again was what we call the father of microscopy, and he invented the microscope. Now, we'll talk a bit more about microscopes later, but just so you know, a microscope is a fancy device that uses different kinds of mirrors and lenses to be able to magnify and zoom in an image. So you can think of it as a complicated magnifying glass. That's really what it is. Okay, so he made an optical microscope, and with the microscope, he looked into many different samples. Um, he looked into samples from pond water, and he saw these very strange little cells that were moving around. He looked into samples from blood, and he saw these different cells that were moving around, and he used many other different kinds of fluids. He was incredibly curious about it, so he tried to use whatever fluid he could get his hand on, essentially. Um, and by any fluid, I mean any fluid. So any fluid that you can think of that comes to mind, he would have tried using that fluid to figure out what was inside it. And what's interesting is back then there wasn't really much transparency in terms of communicating scientific methods. So although Leeuwenhoek made the microscope, he didn't want to reveal how he made it until he took it with him to his grave. It was only his apprentice who revealed it later on, that this was the way that he went about doing it. But he again, people were not really that um, privy to sharing details back then. They wanted to keep things to themselves to have that kind of pride of, oh, I know how to do this and you don't. Now we are in a place that, well, we're in a world where hopefully it's a lot more transparent with sharing scientific methods and ideologies and so on, right? But okay, that's enough about the history. Let's actually look at what he found. Okay, so he found that every organism on the planet is made up of these building blocks, which we call cells, right? These are like single repeating units. So it's a very interesting concept because that allows us to understand certain aspects about life itself better. For example, the reason why a lot of this part of my face looks the same is because most likely they share the same kind of cell, and cells might get a little different as we go along, right? So again, you can define right now a cell as a functional single unit of life. Single unit of life. right? That's not to say that they can't be subdivided. They can definitely be subdivided, and you're going to see that shortly. But you can also combine many cells together to get more complicated designs instead. So just to get some terms out of the way, right? If you have a bunch of cells working together, you then call it a tissue, right? So for example, right now on my face, I've got skin tissue, which we actually would call epithelial tissue. Epi means on the surface, teleal means to do with skin, I believe. So epithelial tissue means, well, that's tissue of cells on the surface of my skin, okay? Um, and a group of those tissues together make an organ, right? And all these tissues have to be working together to make that particular organ. And then again, a group of those organs together will make an organ system. So just to give you another example of this, right? Um, if you've got a bunch of cells working together, that will make a tissue. So for example, in a plant, you could have a single plant cell. A bunch of those together would make a tissue of a plant, which could be the mesophyll layer. That's the layer in the plant that's responsible for doing some amount of photosynthesis, which we'll cover later. And a group of those tissues together could make up an organ, which in a plant could be the leaf. And a group of those organs together could make an organ system, in the case of me, for example, let's say we've got my stomach, my pancreas, my intestines, and so on. All of those will make the digestive system, which is responsible for digesting food. That's the organ system that I'm talking about there. And then a group of those organ systems together, working together, will be making an organism. And it keeps on going as well. So from organism, you can get into more complicated interactions between different organisms. But we'll cover that later in ecology. So for the time being, we'll just stick with this, right? But it just get more complicated. And this is just a really quick misconception. A lot of people think that it should be the same kind of cell that makes up a tissue, but that's not really the case. You can have different kinds of cells that make up a particular tissue, right? So a really good example of this are the cells of the nose, or any kind of surface that you might have in the body that has hair on it. So usually when a surface has hair on it, it has two kinds of cells, the follicle which makes the hair, as well as sebaceous glands or other kinds of mucus secreting cells alongside it. So those are examples of two different kinds of cells which still form the same kind of tissue, right? So you do not need clones of cells to be able to form a tissue. In the same way, you do not need clones of tissues to form an organ, right? So there can be differences between them. But either way, it's just that a group of cells working together to form a particular function is a tissue, and a group of tissues working together to perform a particular function is an organ, and so on and so on and so on, right? So what we're going to look at is we're going to break down what actually is happening inside a cell, just to give you an idea of what's going on here, right? You can find many images of this online, but here's a particular diagram of a cell, okay? And I'm going to just start with an animal cell first, just as a bit of a classic example. Okay, so whenever we call something an animal, it's because it's made up of animal cells. And these are some of the features of animal cells. So let me just draw this kind of blob over here. So this is my weird kind of cell. Doesn't need to be a circle. Most people will draw it as a circle. In reality, it doesn't need to be a circle. It can be whatever shape it wants, as long as it's not too rigid for an animal cell usually. So the main body that you can usually see of an animal cell under a microscope is its nucleus. This applies for any cell, usually. There are exceptions, right? You got to remember that life is very complicated, so there will always be exceptions. But the main part of it will be the nucleus, right? And the nucleus itself is very complicated, which again, I'm not going to go into much detail right now. But this organelle over here, we'll call the nucleus. And I use the word organelle just now to describe one of the compartments that are inside a cell, right? So an organelle is a particular structure that is inside the cell that will be performing a particular function. In this case, the nucleus has its own function. What it does is it actually holds the DNA of the cell. You might hear some people say that the function of the nucleus isn't to hold the DNA of the cell, it's actually to control the cell. But really, that's because the nucleus holds the DNA, which indirectly controls the cell, right? So the nucleus, if you ask me, doesn't control the cell. The nucleus just holds the DNA, and DNA is actually what controls the cell. And we will look more on what the DNA actually is later on. Okay, but again, this is just the nucleus, the first starting point over here. The nucleus also has these funky little holes that let out substances into and out of it, right? So we call those nuclear pores, and this nucleus over here is also having a bit of a membrane of its own, right? Which you can call the nuclear membrane or the nuclear envelope, and so on. Sometimes what can happen, and I think this is really cool, the nucleus can kind of proliferate outwards, right? So if I just draw that over here, it would look something like this. It forms this kind of rib cage of sorts. right? And we call this a reticulum. A reticulum is basically kind of word for rib cage, but it forms this kind of just jet that bulges out in a sense, right? Now this is called the ER, not emergency room, but ER for endoplasmic reticulum. So endo means inside, plasmic means that inside that substance there is plasma inside. So it's an endoplasmic reticulum. And reticulum, like I said, just means a kind of rib cage. Okay. Now the endoplasmic reticulum, this over here has two different functions. The sections which don't have much spots on it is called a smooth endoplasmic reticulum. And the function of this is to make fats for the body or lipid synthesis, right? So the fancy name of fats is a lipid. And the parts that do have spots in it, these aren't just regular spots. These over here are ribosomes, okay? And when you have ribosomes that are dotted onto an endoplasmic reticulum, we call it a rough endoplasmic reticulum. Okay, the rough endoplasmic reticulum just provides a kind of table for these tiny little dots over here to work on. These dots are actually called ribosomes. They don't necessarily need to be float on top of the surface of this guy. They can actually just be floating around in the cell as well, right? So you've got these ribosomes that are just floating around in the cell, and the function of the ribosome, along with the rough ER, is to make proteins, right? So this makes proteins, or you can just say it's protein synthesis. Okay, so you can see the ER is pretty integral to the cell because it makes fats and proteins, and both of those will be used heavily in life, as we will see later on. Okay, and then apart from that, we've also got the another organelle would be the centriole, which looks something like this. It's kind of like a bunch of tubes of fibers that are connected to each other. Again, this is the centriole. This is just an organelle that will help in cell division, which helps to multiply the cell. So it helps in cell division. We've got another structure over here that's quite important, which is the cytoplasm. Now, the cytoplasm is a fluid or it's a gel-like part of the cell, and this is basically providing all of the chemicals as well as the general fluids for any chemical reaction to occur. So we just say that normally the cytoplasm is just the site of all the chemical reactions. Yep. And then we also have this cell membrane over here. Right? Now the cell membrane is quite important, and it's not just because, well, it's not just because it separates the cell from the outside. It's because the cell membrane is actually responsible for controlling what enters the cell and what doesn't enter the cell, right? And you need to understand for cells this is quite important because a cell needs to maintain itself in order to allow itself to continue to survive. If the cell's insides continue to deteriorate, then it is going to stop being a cell, and that is something it wants to avoid in order to continue onwards with life, right? So what a cell membrane will typically do is the cell membrane will throw out any substances that the cell finds as a waste product, and it'll take in any substances from the surroundings that the cell finds as a useful nutrient, right? And this can be modified in many different ways depending on the function of the cell. But again, that is the purpose of the cell. So one is that it actually provides a barrier between the outside world and the inside world of the cell. And the other is that it also controls what substances are entering into the cell or out of the cell. So again, we can say that this controls the movement of substances into and out of the cell. So we're just going to say movement of chemicals in and out of the cell. Right, so that is what the cell membrane over there is doing. Okay, now apart from this, there are also some other organelles as well. This one's very famous, right? So it's not actually this large. In reality, a lot of the stuff here that I'm drawing here are not to scale. If I had to draw this to scale, you wouldn't be able to see it. So this organelle over here, if this was to scale, would actually be this tiny. It would just be a speck. It would be a bit of a dot. You can't really see it. So I'm just drawing it a bit bigger here to simplify the diagram, right? But anyways, this over here is an organelle that looks a bit like this, and it's got this kind of wavy lines within it, which I believe we call cristae, right? But this over here we call the mitochondria. And as you may or may not know from all the beams and so on, we call the mitochondria the powerhouse of the cell, right? But really what it does is it helps produce energy for the cell through a process that's called aerobic respiration. And we'll learn more about aerobic respiration later on. But essentially, in a nutshell, that is a type of reaction in which the cell will make an abundance of energy, right? So again, the mitochondria is the way for the cell to make a lot of energy. So this again is you can say it's the site of aerobic respiration, right? So again, it's a site of aerobic respiration, which indirectly means that it produces a large amount of energy for the cell. Yeah, so I say that it produces energy, but really what it does is it produces ATP, and you can think of ATP as a type of chemical that is the energy currency of the cell. So the cell uses ATP as a currency to do different things, whether it's to make proteins, or whether it's to make fats, or whether it's to move things into and out of the cell. That is what ATP, which is produced from the mitochondria of some places, is used for, right? But remember again that this is all very versatile. There might be cases where in terms of the word aerobic over there means that there needs to be oxygen present. If there's not enough oxygen present, then the cell needs to find a way to get energy without any oxygen present, in which case the respiration process will happen in the cytoplasm instead, right? In which case we call it anaerobic respiration. So that happens in the cytoplasm. And remember, anaerobic respiration just means the absence of oxygen. Typically, it's more preferable for complicated organisms to perform aerobic respiration rather than anaerobic respiration. The only reason why something might be doing anaerobic respiration is because there's either not enough oxygen or because the organism is simple enough, like a bacteria, to be performing anaerobic respiration, right? Um, okay, right, but these are the main points. And again, I've only drawn ribosomes over here, but they can be sprawled out throughout the cell, like this. Okay, so just moving on then. Okay, so this is the general structure of an animal cell, right? And again, there's a lot of stuff that we're omitting. For example, we're not mentioning the cytokinetic skeleton, or the cytoskeleton, I suppose. We're not mentioning the temporary vacuoles and so on. There's a lot more detail and nuance to this. But what I want you to just keep in mind is it's this over here is just a general kind of template for what a cell could have. It's not necessarily always the case that every animal cell will have these features in them. What a cell has usually depends on its function, right? So there are times where a cell might not even have a nucleus, for example. It really just depends on the function of the cell. So just keep an open mind as we're learning about the different things in biology. This is over here is just kind of like the average unit of what you would expect to see within some randomly picked cell, right? But anyways, this over here is what it looks like for an animal cell. And you've also got another kind of cell, which is a plant cell. Okay, now I'm going to omit all of the common things I've drawn just now, which is the nucleus and the mitochondria and so on. But I'm just going to highlight what is the main feature of a plant cell. Now, a plant cell has this outer covering that we call a cell wall. And the cell wall is very important for a plant. Okay, so the inside membrane is just called a cell membrane, but the outside is called a cell wall. Okay, so a cell membrane is made up of a very smooth, watery, fleshy kind of substance, I suppose. You will learn more about that later on. It's called a phospholipid. But a cell wall is not made up of that kind of smooth substance. It's made up of something that is a bit more tougher, more rigid. And what it's actually made up of depends on the plant and the location of that cell. But typically, it's made up of something that's called cellulose. Okay, all you need to know about cellulose for the time being is that it's very rigid in structure. And the reason why I'm emphasizing the fact that it's rigid is just because the cell wall is very important towards holding the plant's solid structure in place, right? The reason why plants are very rigid compared to us, for example, if you feel a piece of your own flesh, right, that is just going to be, your flesh will feel soft, right? That's the characteristic feature of an animal. Um, the they are softer, and the only parts where they are tough are maybe at the bones, right? But if you look at a plant, plants are way tougher than animals tend to be, especially when they produce these tougher tree barks and so on, right? Now, the source of that bark and what's responsible for keeping the plant upright and maintaining that structure is cellulose, right? It's that hard part of the cell wall. So this over here provides that rigidity to the cell, okay? And the way that it does this is quite ingenious. Again, we'll learn about this more later on, but in a nutshell, the way that this works is what the cell wall does is it allows the plant cell to take up an abundance of water, right? So once the plant cell takes a lot of water, it's not going to get to the point where it's going to burst anymore. It's just going to maintain all that pressure within the cell itself. So if an animal cell took up too much water, then the animal cell would actually end up exploding because the cell membrane would not be able to contain it. It's not strong enough. But a cell wall would actually be tough enough to hold that plant in place, right? To hold that structure in place. So that is the key point about a cell wall. It's quite important for it to be able to maintain the structure of a plant. This is again the reason why if a plant was not given enough water, it's going to wilt because the cells are all going to shrink. The pressure within them is going to subside. There's not enough water in them. But if you give it enough water, that pressure is going to rebuild itself again. So that has a name. It's called turgor pressure. But we'll just go back and focus on the organelles for the time being. So the plant cell has a nucleus and it has mitochondria of its own. It has centrioles as well, and so on. But what it also has that that an animal cell doesn't have is that a plant cell has a giant permanent vacuole. Okay, and it's permanent because this is opposed to animal cells which have temporary vacuoles every now and then. Now, a vacuole is a particular membrane-bound storage area of a cell. In the case of plants, it stores something that's called cell sap. Cell sap is just a solute. Solute just means that it's got some amount of water that's been dissolved in, or that's dissolving some amount of fat, or not fat, sorry, but salts, right? But in this case, it's a solute of salts and glucose. Okay, that's typically what a cell sap contains. It might contain a few other things like ions, but this is typically what it contains, right? So the permanent vacuole over here will have this cell sap in it, and this is just meant to be that excess water supply for the plant in case it needs a bit more pressure, right? The other thing that, or well, the most important thing of a plant, if you ask me, is this little organelle over here, which again is as small as mitochondria. So realistically, if I drew it, it should be small like that, right? But this organelle over here has a telling feature, which includes these kind of plates. They look like plates of sorts that are stacked on top of each other. We call them grana. But for the time being, you should just know that this is called a chloroplast. And a chloroplast is responsible for photosynthesis. Photosynthesis is the way by which plants make their own food. And we will go into this in more detail later on, right? But that is what a chloroplast actually does. It is responsible for the plant conducting photosynthesis. It's also the reason why plants are actually green in color, because chloroplasts have a pigment in them that makes them green. That pigment is called chlorophyll. But a plant cell doesn't need to have any chloroplasts. Again, remember that life is very complicated, so it can adapt and change itself depending on the needs of the system. So, for example, rather than being a chloroplast, it could also be something that's called a leucoplast. Now, the word chloro means green, by the way. Leuco means white. Plast means body. So you can see they were very original with names. Chloroplast is just green body, right? It comes from Latin. So, anyways, the next one is a leucoplast. And this over here is a white body. So what does it have? It has potato chips in it. Not literally potato chips, but it has starch, which is what potato chips have in them. And the reason why they're white in color, if you've ever cut into a potato, that's the same kind of color you can expect. And that's the reason, again, why they call it a leucoplast. It's that white in color. That's coming from it storing starch. And starch is just the compound that's found in potato chips and some other plants and so on. It's meant to store energy, right? And then you've also got chromoplasts. So chromo means color. So chromoplasts are what are responsible for giving plants color. And we're talking about not just green, but different colors. So for example, you could have an apple. The surface of the apple is originally green because it has chloroplasts on it. And eventually, once it becomes red, that's because the chloroplasts are now slowly turning into chromoplasts. The and if you're looking at a potato, for example, if you ever take some potatoes and you store them in sunlight, you will see that the top of the potato surface actually turns green in color. That's because the leucoplasts of the potato are slowly turning into chloroplasts, and that will slowly develop into its own potato plant, right? So again, life is very versatile in this kind of aspect. Now, that is what an animal cell looks like up there, and here is what a plant cell looks like. So you can see there's a few differences between them. Now, the other kind of cell that I want to just go through really quickly is a bacteria. There's not too much you need to know about a bacteria, but I'm just going to highlight the main details. Okay, so a bacterial cell can look something like this, right? So there's many different kinds of shapes of bacteria. For example, you've got circular ones, which we call coccus. We've got these comma-shaped ones. I think these are called vibrio. I don't fully remember the name, but I believe those are called vibrios. And then you've got these ones as well, which are long ones. You've also got these really fancy ones. I think they're called spirogella, but they spiral down like this, so really, really cool. But either way, we're just going to focus on the typical kind of bacteria cell, which looks like this. This is from a strain of bacteria called E. coli, a very common bacteria that's responsible for stuff like diarrhea and so on. Okay, now this kind of structure over here, I'm kind of drawing it for exaggerated effect, but bacteria can have, they don't need to, they can sometimes just have only 2 layers, but they can have 3 layers. The inner layer is again the cell membrane, identical to what we discussed earlier. And then we have the cell wall, and then we have over here the slime capsule. You can call this the slime layer or the capsule layer, or if you want to be somewhat fancy, you can call it the slime capsule. Um, the function of the cell wall, as you know, is to provide that strength to it, right? That rigidity to it. The function of the cell membrane is, like before, to control the substances moving into and out of the cell. The function of the slime capsule is to just provide an extra layer of protection from the environment in case anything is trying to affect the bacteria, right? Apart from this, bacteria can also have these extensions over here, which we can call flagella. These are hair-like substances which are responsible for a swimming motion. But we mostly say that this is for locomotion. But I'm going to instead use the word movement. Okay, so locomotion, that term in biology, locomotion, is usually used for a more sophisticated process where something is using a series of steps to move from one place to another. For example, humans, right? When humans are walking, we are using all pairs of our muscles in our legs, right? And we're walking in very complicated ways. So that would be described as a locomotion. But a movement could just be like me moving my hand, right? So if I'm moving my hand right now, this would be called a movement. But if I am moving my entire body, walking from, I don't know, where I am right now to the grocery store, then you'd call it locomotion, right? So in the case of bacteria, I would be more inclined to call the flagella's movements over here just movements, because flagella don't actually move themselves in a very sophisticated way. They just randomly cause the bacteria to bump all over the place. So the bacteria kind of just randomly moves around, right? Um, so you have to understand the reason why this is the case is because in a bacteria, the cell is very tiny. It's not as complicated, nor is it as sophisticated as an animal cell, which is way larger. Just to give you a scale of reference, right? An animal cell might be that big. A bacteria is right over here, okay, or maybe even smaller, that size. Which, by the way, is coincidentally matching the size of what a mitochondria looks like and what a chloroplast looks like. And it's not, well, we have theories as to why that's the case, which we might visit one day. But otherwise, for the time being, again, just keep in mind the scale difference, right? So a lot of my diagrams are exaggerated. Just keep in mind that this is we're talking about really tiny scales right now, scales to the point where we cannot see it with the human eye, so we need to rely on using a microscope. But okay, so we've got this flagella over here that beats the bacteria against the water in many different ways to just cause this random kind of movement. And then within the cell, within the bacteria cell, a very interesting thing about it is that it doesn't have any membrane-bound organelles. So if you remember from what we drew earlier, we had the animal cell and the plant cell that had a lot of these membranes that were binding their organelles in place, right? So it made a kind of cell within a cell. It's like a bit like inception. But bacteria don't have that. They don't have this double kind of structure of sorts, right? This compartment. They don't have that compartmentalization. Um, so what they have instead is they have these free-floating organelles instead. Okay, so just some of the key features are the DNA of the bacteria. For us and plants, the DNA is usually linear, so it starts like this and it winds around like that. That's what the DNA looks like. However, in a bacteria, the DNA is actually in a loop. We call this loop the nucleoid. And there are some more loops of tinier pieces of DNA in the bacteria, which we can call plasmids. So these are circular rings of DNA. These become really important in genetic engineering, and the bacteria actually uses this to transfer genes from one bacteria to another. So, for example, if one bacteria is resistant towards an antibiotic, then that bacteria will try to share the plasmid to another bacteria. But there are some things that the bacteria can do with the plasmid that get really weird, really trippy. For example, one of the things the bacteria can do is it could purposefully share a plasmid that will help destroy any bacteria that are around it. And the reason why is to decrease the amount of competition so that this one particular bacteria can survive on its own, right? So that would decrease the competition. But again, this is just a circular ring of DNA that's called a plasmid. So that is a circular ring of DNA. And again, as you'll see, it becomes important in genetic engineering. Now, these bacteria also have ribosomes in them, but there is a very small difference to the ribosomes of what we drew above. The ribosomes over here are slightly smaller, so we call these 70S ribosomes. So these are slightly smaller than the ribosomes of the cell above. For the cells above, the ribosomes are instead 80S. Okay. In case you're wondering what an S means, an S stands for 1 Armstrong. An Armstrong is a unit of 10 to the power of minus 10 meters, and that is it's approximately the size of an atom, right? So that means that the ribosome is basically a diameter of 80 atoms in length in this case, whereas for the other one it's slightly smaller, so it's simpler, and it's 70 atoms in length in terms of scale, right? So you would need an incredibly sophisticated and complicated microscope to be able to see a bacterial cell. Okay, now this is a bit of a rundown on what cells are made up of, but you also just need to understand that there is a bit of division of cells, and you can categorize cells based on their similarities and their differences to each other, right? So what I'm going to show you now is a concept of something that's called the tree of life, where we have organized the different kinds of cells and species and organisms and so on based on their similarities to each other, right? And we will learn more about this when we're going to ecology and taxonomy later on. But just to give you a bit of a rundown, right? When we're talking about life on the planet, life can be categorized into what we call eukaryotes and prokaryotes. So eukaryota and prokaryota. Now, the EU part in eukaryota means new. So, and karyote, by the way, just means carrier or cell, right? So eukaryotes are the new cells. These are the newer versions of cells that came up later. So we are comprised of these newer versions, right? So again, this is like the newer versions of cells, the more sophisticated ones. Whereas prokaryotes, those are the ancient, older beta versions, if you will, right? Those are the undeveloped like versions of cells. Not that they're undeveloped per se, but it's more so that these cells came first. In fact, the word pro means professional, or if you say that someone's a pro at gaming, it means that they're at the top, right? So pro means first. Okay, so prokaryota means it's the cells that came up first, and whereas eukaryota are the cells that came up only after some time, relatively speaking, compared to prokaryota. So, prokaryota can then be divided further into archaea and bacteria. Bacteria, what we just talked about just now, archaea is similar to bacteria, but there are a few structural differences between them, which we won't go into for the time being, because the main one that I want you to know about is just bacteria. And then for eukaryota, these can be divided into further categories as well. We've got animal cells, we've also got plant cells. These are the ones that I covered, but you can also classify them into fungi. And you can also classify them into, what do you call it? Protoctista, right? The fancy name for animals is Animalia. I think for plants it's Plantae. But anyways, right, I'm just going to give you a brief rundown of what these different categories are, right? So when you think about Animalia, these are usually multicellular organisms, right? So they are multicellular organisms, meaning that they're made up of more than one cell at least working together, okay? And these have animal characteristics. So this typically means that they're made up of organisms that try to eat other organisms. They typically get their food from other organisms, right? So again, when we talk about Animalia, they are heterotrophic. Hetero means different, trophic means food, right? So they get their food from other places. They're heterotrophic, and they are also, what do you call it? These include things like fish, insects, mammals, reptiles, and so on, right? So all of these fall under Animalia. Now for plants, those would be trees, shrubs, grass, and so on. And these over here are autotrophic or phototrophic. Auto means same, and trophic means food. So it uses just itself to get food. And plants can get their food from just sunlight. So what autotrophic basically means in this context is that they are independent. They are not reliant on other organisms, in a manner of speaking, with just food, right? They're not reliant on other organisms. Now, fungi, these over here are defined by the fact that they are actually decomposers. So they tend to feed off of different things. They tend to be decomposers. Not that they have to be. There are a few exceptions to this, but they tend to be decomposers, meaning that they feed on organisms that are already dead, whereas animals might feed on organisms that are still alive, or they might kill an organism in order to get the food from that organism. Whereas for a fungus, it's not going to kill an organism per se. Like some of them might, okay, but generally speaking, most fungi will just wait until the organism dies, and then it'll do the dirty work of just cleaning it up. It doesn't, bother to get its hands dirty. Now, the telling feature of a fungus compared to an animal cell is that a fungus has a cell wall. And the cell wall is different to plant cells because in a plant cell, the cell wall is made up of cellulose, whereas in a fungus, it is made up of something that's called chitin. Chitin is actually more similar to insects than it is plants. Therefore, what that means is animal cells are actually more similar to fungal cells rather than plant cells. So a lot of people think that fungi are more related towards plants. Really, they are actually close relatives to us, which seems weird, but that is the biology of it. And then finally, we have the Protoctista, my favorite kingdom, because of how often it gets forgotten. And we call this the Dustbin Kingdom. That's the nickname for it. The reason being that essentially, if you've discovered a new organism and you're struggling with trying to put it into the animal kingdom or the plant kingdom or the fungal kingdom, you have no idea which one it falls into, then just chuck it into the Protoctista kingdom. That's the reason why we call it the dustbin kingdom. It's because if you don't know what something is, just toss it in there, right? So Protoctista tend to be unicellular organisms, right? That means that they are made up of just one cell. So an organism that is made up of just one cell doesn't need to be, but it can be just unicellular. And any organism that has traits that are mismatched, that doesn't make it clear as to whether it's a animal cell or a plant cell, would fit into the Protoctista category. One of my favorite examples of this is Euglena. And Euglena looks something like this. Kind of looks like a bit of a mouse. It has this tail over here, this long flagella that it can use to move around. So this is a kind of cell, okay? The thing that's interesting about the euglena is that it can go ahead and it can actually eat other cells. However, it also has chloroplasts in it, which can also be used to make its own food. So an euglena is a bit like an outlier, right? So it can make its own food, so it's autotrophic, but it could also eat other organisms, so it can also be heterotrophic. Usually what it is depends on the type of species of euglena, and it also depends on the environment the euglena is in, right? So, for example, if it's around a lot of cells, it might try to adapt to become more uh, what do you call it? It might try to feed on more animal, like more cell surrounded instead of synthesizing all the time, I suppose. But that is what an example of a Protoctista is, right? So just keep in mind that these are the four categories of eukaryota. Now, for prokaryota, those could be archaea, or they could also be bacteria. Archaea the name of archaea itself comes from archaic, which means old and odd, I suppose, or old and ancient. So these came pretty. Those were one of the first organisms to arrive. Essentially, that's what we believe. And for bacteria, those are ones that we see in abundance all around us. They are all over the place. So you've got many different kinds. You've got bacteria which come up in rods like this. You've got bacteria which go in spirals like this. You've got bacteria which are just in these little circles like this. And you've got different bacteria that are kind of like in comma shapes like this. Okay. Now an example of a bacteria could be E. coli. E. coli is used for a lot of different things. Like, for example, it is used for making yogurt, but it's also the same kind of bacteria where different species of it could give you food poisoning, for example. A lot of bacterial diseases come from, well, a lot of diseases come from bacteria. Okay, now some of you who are very astute might be noticing that there is one group in here that I haven't written about at all, and that group actually is a virus. And some of you might also be thinking that why am I not connecting this towards life? It's because what a virus actually is up to a lot of debate, or what it's actually made up of leads to a lot of debate as to whether it's considered alive or not, right? So just to give you a bit of an understanding, in order for something to be considered alive, it needs to be able to divide itself on its own. However, a virus is incapable of doing that on its own. It also lacks the distinct features of the cell. So just to give you an idea of, of what a virus actually has in it, right? A virus can be composed of an outer envelope of a cell membrane that's not even its own. It comes from the host cell that it steals it from. So it's a stolen host cell membrane. Inside this envelope is a thin layer of something that we call a capsid. A capsid is just a protein membrane, and inside that capsid we have a bunch of DNA or RNA in it. It depends on the type of bacteria. Virus. But HIV, for example, would have DNA in it. Whereas other viruses like bacteriophages could just have RNA in it. RNA is another kind of chemical that again we will learn more about later. But in a nutshell, this is what a virus is, and it's a very simple particle. Okay, I would not call it an organism. It is more of a particle. And again, that's not me saying that it isn't alive. It's just most of the fact that it is definitely much more different to what we would consider animal cells and plant cells as being, right? And the reason why it's a lot more different is because a virus really only shows signs of being alive in a sense once it's inside the host cell that it's trying to infect. The way that the virus replicates itself and the way that the virus reproduces is by infecting a host cell, ordering and hijacking the host cell to make many copies of the virus, and then the host cell releases all of these viruses and dies in the process. That is essentially how a virus works. That's also part of the reason why viruses are really hard to treat, right? Whereas bacteria can be treated with antibiotics, viruses are way smaller. And again, just to give you a frame of scale here, remember that if this was an animal cell, then a bacteria would look like this. In the same way, if this was a bacteria cell, then a virus would look like this. Okay, so again, bacteria over here, virus here, animal cell over here, bacteria here. So viruses are incredibly tiny, and we only discovered about them much later. Okay, now with that said, remember what I've said about science in the beginning? Science is trying its best to be as objective and as quantifiable as possible, right? So when we're talking about life itself, what is considered alive is kind of a murky area, right? Do you consider viruses alive because they can infect other organisms, or do you not, right? So in biology, in order to quantify what is considered alive and not alive, there are some key characteristics that they need to follow in order for us to consider alive, right? So again, this is to do with life itself. Okay, and right now we're talking about the characteristics of life. Okay, so for the characteristics of life, one of the key things that they need to be doing is they need to be capable of growth, right? So anything that's considered alive should be capable of growth. The other thing they should be considered capable of doing is being able to reproduce on their own. They should also be capable of producing energy for themselves, which we call respiration. And viruses, by the way, don't do that, right? They also need to be showing signs of excretion. Excretion is a chemical process that releases waste from the body, right? So this is basically the removal of nitrogenous waste. Nitrogenous waste, this means that it is any kind of waste that has nitrogen in it. And respiration is, again, it's a process by which the cell will produce energy, right? The other point as well is that it needs to show some kind of a nervous system, which is not necessarily the case because, of course, for a plant cell or plants, they don't really have a nervous system, but it should be some kind of way of showing response to stimulus. That's essentially what this means. A stimulus is a change in the environment. So for example, if I poke myself, that is me giving myself a stimulus. It's a stimulus because it's a change in the environment and my body is able to detect that change. And if it really annoys me, I can make my hand stop, right? So that's a response to that stimulus. And this is typically what organisms should have. So again, they should be able to grow, they should be able to reproduce, they should be able to respire, they should be able to excrete, meaning they should be able to throw out nitrogenous waste, and they should be able to respond to stimuli, or in other words, they should be able to have a nervous system. These are the key qualities of life. And going back to viruses, viruses don't have that at all, right? So viruses are not able to reproduce on their own. They can't even grow on their own. They cannot respire. They like literally cannot, and they don't have the organelles or chemicals for it. And they don't really undergo excretion. They also do they even respond to stimuli? So it's a little weird, right? So viruses are dormant and inactive for the most part. So usually when you see a virus under a microscope and it isn't moving or it's dormant, we call it instead a virion. Okay? And it gets even weirder than that, right? So remember that viruses are kind of like borderline to life, I would say. Like you're not really sure about whether they are alive or not alive. In the same way, you've got another kind of substance that's called a prion, right? So a prion is basically, I guess the simplest way I have of explaining it is you can think of it as a type of chemical that when it infects the body and it finds similar chemicals, it turns all of those chemicals into itself, right? So again, just to reiterate, a prion is a type of chemical, it really is a protein. It's a type of protein where when it finds another protein that isn't quote unquote infected, it infects that protein by turning that protein into itself. And then that protein will go on to continue that quote unquote infection, right? So a prion can also cause diseases as well. And this can cause, for example, mad cow disease, which is when a cow basically goes rabid, right? But that is caused by a prion. So you see, in terms of biology, what actually is considered alive is kind of it's quite philosophical in a sense. Like, would you consider prions alive? Would you consider virions alive? At the end of the day, regardless of what you think of it, just know that science tries to quantify what we consider alive, and it quantifies life as um, having one of these traits. Okay, so I remember I was once asked a question. It was a really nice question. And what I was asked was, does a cell need to have DNA in order for us to consider that it's alive, right? But that's not really what the student meant. What the student actually meant was if you find a cell at another planet and it's got a completely different like biochemistry to our own, would we no longer consider it alive? And again, that goes more into the philosophy of like, okay, what do you consider alive, what do you not consider alive? But again, in biology, these are the key points. If that cell that we found, an alien cell from another place, that's got completely different biochemistry, in order for biology to consider it alive, we would have to say that it's showing evidence of being able to grow, it's showing evidence of being able to reproduce, it's showing evidence of being able to produce energy in some way, whether it's eating other things or just making it itself, and it shows evidence of being able to throw out any kind of waste material, and finally it is showing evidence of being able to react to changes in its environment. Okay, those are the qualities needed for life. So for example, there's a bit of debate about whether AI could be considered alive. In terms of biology, AI would never be able to be considered alive unless that AI was capable of showing growth, which I'm not sure how you would define that, or it's capable of replicating itself, which could count as reproduction, it's capable of producing its own energy, which maybe it can't by taking electricity, and then it's also capable of, I don't know, throwing any waste it might have, right, and reacting to its environment. By that definition, AI would be considered alive. Keep in mind that this is different to me saying that something is conscious, right? So consciousness is something that's not fully understood just yet. But when we're talking about consciousness, it means we're talking about an ability to be able to be self-aware, right? Being able to be sentient, I suppose. Sentient is a better word for it. So being able to be self-aware, being able to have the realization that you are someone who is alive and you're in this world of people who are also alive and you are different to what is around you, right? For example, I myself, I know that I'm alive and I know that my hand is different to my environment. My hand is a part of me, right? So that is what sentience is in a sense, right? So when we're talking about life, do not conflate it with sentience. And we're not talking about whether something is considered to have a sense of itself or anything like that. We're just talking about whether something is considered to be a living thing or a non-living thing. A non-living thing would be something like this pen right here, which is just there. Okay, right. So those are the key points about cells. Now, just to move on. All right, so just drinking some water, but just to continue, right? So just to summarize what I was saying, with a root hair cell, that is an example of a differentiated plant cell because it's much more different to a regular plant cell because this has no chloroplast, because it doesn't conduct photosynthesis, doesn't need to because it's underground, so it doesn't get sunlight for photosynthesis. And it also has an appendage that's long over here to be able to absorb nutrients from the soil, right? And just I've also drawn two other examples of differentiated cells. So these are examples of animal cells which are differentiated. So you can see over here that this is a nerve cell where the main body of it has a star-like shape and it's called the cyton, whereas the tail end of it is called the axon. And the function of a nerve cell is to just transmit the electrical signals from one area to another. That's the point of it, right? Whereas in another kind of cell, this is an RBC, right, or an erythrocyte, this is a special type of cell that's found in the blood, and its main function is essentially to carry oxygen around the body. That's what it does. So the thing that's interesting about this is that red blood cells originally, when they are first made, they actually have a nucleus in them. But as they mature, the nucleus actually dissolves away and they get rid of the nucleus. That actually causes the middle part of the red blood cell to have a dimple kind of hole. So if I drew a cross section of red blood cell, it would look like this. Think of a donut, right? Just think of a donut, but imagine that the inside has like this connected layer between them, right? So that's what an erythrocyte looks like. It has this biconcave disc-like shape, and it's packed with this protein that's called hemoglobin, which helps it carry oxygen around the body, right? But again, that's an example of a cell that is differentiated, and it's also a good example of that golden rule that I mentioned, where it's always the cell that modifies itself to fit the function, right? Remember, the labels, the categories that we make are made up. Okay, when I say that something is an animal cell, when I make a category of animal cells, I'm trying to just do my best to simplify the world around me. That doesn't mean that the world works in that way. Okay, so in reality, cells don't care about what you categorize them as. They will tend to do what is needed to be able to adapt for their environment. That's how it goes, right? That doesn't mean that they can adapt super quickly under a small amount of time frame, but over a course of many generations, they might just be able to. Okay. So again, an erythrocyte is a good example of that because it goes against that rule of animal cells always needing to have a nucleus. It's not always the case that animal cells need to have a nucleus. There are times where they don't, as you can see over here, right? But that again is an example of differentiation and specialization in cells, where cells can differentiate to be able to perform a particular function from a kind of blank template cell that doesn't have much features at all. Now, usually, okay, most of this differentiation, as I've mentioned right down here, happens when the cell is a zygote, meaning at the embryo stage, when a person is still growing, and we're talking about growing in the sense of them being super young, still at the point of conception, right? That's when differentiation tends to happen the most. As you grow older, your cells lose that ability to be able to differentiate more and more and more. Okay, so we call this differentiation, specialization, and we also call it the ability of a cell to be able to divide into many different things is called totipotency. So potent means, like the ability or the strength. Toti means everything. So totipotency means it's a cell that that can specialize into many different kinds of cells. So it can spell that it can what do you call it? So it's a cell that can specialize into other kinds. Okay. Not every cell can. For example, with us, only our stem cells, or which we call embryo cells, can be totipotent. Another word for totipotent is pluripotent. There's a slight difference between them. Totipotent just means it can divide into absolutely everything, right? Whereas pluripotent means it can divide into some things. Okay. Now, there's also another one which is unipotent, or there's a specific name for this, but we'll just call it unipotent for now as a bit of a placeholder. But certain kinds of cells can divide into just themselves, right? A really good example of this are the hepatic cells, which are the liver cells. So it can divide into just itself. Divide into just itself. So our liver, for example, can do just that, right? So the liver is an organ that gets stressed really easily in the body because of the sheer amount of functions that it has to do. So liver cells often need to be able to regenerate to replenish themselves. There are other organisms in the body that cannot divide themselves at all. For example, the brain. Once the brain is fully developed after the age of like 25 is estimated, once the brain is fully developed, it will no longer be able to divide. Now, some people call this neuroelasticity and neuroplasticity and so on, but again, you can just know that in terms of the brain, once it's formed, will no longer make any new cells. So that's why it's very important to try and just continue training the brain after that age threshold of 25 to be able to make sure that you don't lose that IQ, because that's how IQ usually goes. It increases all the way to that age, but then up that age, it starts to steadily decline unless the person will regularly train the mind or do other kinds of IQ tests and mental like exercises, I suppose. All right. So apart from that, I'm going to just very briefly talk about microscopes. Just to circle back to where we started from the beginning, right? So when we first introduced you to this, I was telling you that Robert Lee Van Hook discovered about the wonderful world of cells by making a device that's called a microscope, right? The way that the microscope works actually works from physics. It works on physics principles, but the way that you can think of it is you can just think of two magnifying glasses that are doubly magnifying to be able to zoom in on a material. Okay, so because of that, we have two different kinds of microscopes that we use today. One that we call an optical microscope. Optical just means light. So another word for optic microscope is a light microscope. And this again uses magnifying glasses, but we don't call them magnifying glasses, we call them convex lenses. So they use convex lenses to magnify the image. And the nice thing about this is that it can produce colors of images, but the resolution is lower compared to other things. Okay, now the other thing that I'm talking about is an electron microscope. An electron microscope doesn't use light to make images, it instead uses electrons. And there is a reason why this produces higher resolution images, which I can't explain just yet, but we will explain this once I get into physics later on. This would have to be in high school physics. But for the time being, the way that you can understand this is just that electrons will, can fit through tinier gaps. That's how you can think of it. It depends on the speed of which the electron is shot, usually. If it's shot at a high enough speed, then it can fit through tinier gaps. But light is not the same. The light that we can see has a fixed wavelength. We cannot see any further than that wavelength or any shorter than that wavelength. We don't have the eyes to be able to see that, right? So although it is theoretically possible to use light of lower frequencies like UV rays and X rays, the problem with that is that those would just pass straight through the cells. They wouldn't even reflect off of the cells. So electron microscopes are pretty good in that regard. Okay, so again, electron microscopes use electrons to produce an image. Now, the benefit of this is that it produces a higher resolution image. However, the image is going to be monochrome. And the other downside as well is that this will most certainly kill the organism. So if you're wanting to get a live view of something, to see what something looks like under the microscope, for example, if you wanted to look at a Daphnia, which is a type of water flea, and you wanted to see its heart literally beating under the microscope, you would only be able to do that with an optical microscope, which can see light. Because light is not going to kill an organism, typically speaking, right? But an electron microscope would have to kill the organism usually because the organism is basically being bombarded with electrons, and those electrons are going to ionize the cell and kill it essentially. Um, okay. Now, apart from this as well, yeah, apart from this as well, I suppose the last thing that I will just mention, is just a bit of outlier points, just so you're aware of it in the future. Um, it is possible for us to culture organisms. Okay, culture just means a growth of something. Okay, so in normal English, culture means the background, right? But I guess in this context, culture is referring to a particular growth of something. Usually, it's a growth of a singular organism, right? So as biologists, we can culture different organisms. Some of those organisms, for example, could be bacteria, right? So if you have a single cell of bacteria And if you have a Petri dish is a type of glass dish. So if you have a Petri dish and you sterilize a Petri dish, sterilization just means you're killing off anything else that's in it. Usually this can be done with alcohol wipes or UV rays. What you can then do is you can then introduce a single cell of that bacteria onto some kind of food for the bacteria to eat. For example, it could be agar gel. Agar gel is a type of jelly which has nutrients. It's a kind of broth for the bacteria to basically feed off of, right? But you could basically make the bacteria feed on this agar gel, and this will give you a single culture of nothing but that particular bacteria. The reason why you might want a single bacteria to be duplicated that much is because maybe you'd want to study what happens to a section of that bacteria without ruining your sample, right? Or maybe you'd want to study what happens to that bacteria when you're introducing it to different kinds of antibiotics. So it really depends on the context, right? But this is just an example of us culturing an organism. You don't need to culture only bacteria. It's also possible to culture plants. People do this in tissue culture. It's also possible to culture fungi as well. And it's also possible to culture animals as well. So you could take a type of animal cell and you could grow it into fully fledged organs. But there are some ethical controversial issues that are around that practice, right? But it is something that people are capable of currently doing. But again, right now, the current climate is that it's being held back. Um, but yeah, these are the different main points, I suppose. Right? So what we'll do for next time is we will go back to cells and we will look more on how our cells are actually capable of dividing. What's the actual process of cell division? Right? So that's what we'll look at. Right? So just to summarize, remember that a cell is a single functional unit of life, and it's not as simple as every cell is made the same. Cells are heavily differentiated based on their function, right? That's the main point I want you to take away from this. So that's the reason as to why life itself is very complex. It's because cells themselves can be very complicated in their design, and they can adapt themselves based on the function that they need to take. Adapting themselves doesn't necessarily mean that they do it in one generation. It might mean that they take multiple generations to be able to do so. Or if they already have the data in them within their DNA, for example, then they could specialize into something else. The other point as well are the domains of life, right? So life itself is divided into two domains, which we call eukaryota and prokaryota. Eukaryota can be divided into these further kingdoms here, and prokaryota can be further divided into these kingdoms here, right? So that is us arbitrarily trying to categorize life. But again, life doesn't care about whether we are categorizing it or not. Life will just do what it wants to do, right? So all we're doing is we're observing what happens to it. Okay, so again, for the next session of biology, we will look more at cell division. But the next slide that I will release, the next lessons that I will release will be more in chemistry. And after that, we will go into physics and we will keep on cycling back like this until we are done with middle school biology. But yeah, I hope you enjoyed that. Let me know if you have any questions, and thank you for watching.